Good afternoon, everybody. My name is Benjamin Coltman, and yes, I'm a PhD student who's had the real joy for the last 4.5 years to work at Boku University in Vienna, specifically in the CD laboratory for growth decoupled protein production in yeast, working with Karina Rebneder and Brigitte Gasser, as well as a bunch of other lovely people. And today I'll be talking about long non-coding RNAs, and I will dive straight in. With what are they? They're transcripts, they're non-coding, and they are over 200 nucleotides, and that is the end of my presentation. They are transcripts. They might have a poly-A tail. They might contain introns and be spliced and therefore be multi-exonic. I really should have removed. Their 3D structure is often important and defines their function. Typically, they are classified with their proximity to the nearest protein-coding gene, in which case they might be antisense, where they're on the opposite strand to their nearest coding gene. There might be sense, where they are fully contained within different genes, or they might be intergenic, where they are outside of the other genes. How do they function? They have a very, very, very large interactome, which is really quite hard to try and probe or even understand what they do. But they might interact with proteins. They can recruit and decoy chromatin modifiers. They can interact with peptides, which might lead to assembly of nuclear condensates. They can directly form triplexes with DNA, which might therefore lead to gene silencing and activation. They can directly interact with possibly small molecular weight compounds, which may modulate their activity. They can interact with other transcripts, which might modify the stability of both the long non-coding RNA and the transcript, and it can also lead to their differential localization. So they can do a lot of things, and it's really quite hard to know what they do. But in the CD laboratory, we're particularly interested in glucose-limited slow growth rates. One, from a basic biology point of view, just to understand what's happening, but also to guide strain and process development. So I'll just take this second to plug that I've got two other colleagues that are presenting posters. So Jennifer Staudiger will present as a poster presentation 2 different strategies for improving recombinant protein production, and Karina Rebneder is one of the other rare virtual posters that is looking at improving a promoter which is glucose limit induced. But more back to sort of this presentation, long non-coding RNA expression levels are typically associated with stress. And one source of stress might be just changing growth rates, which is where we come in, and where I wanted to sort of question, can we identify long non-coding RNAs? Are they associated with substrate-limited near-zero growth? And can we determine the function of that very large interactome? Is it possible to get to a solution? And could they potentially be good engineering targets? So I'll just explain near-zero growth rates because it might not be clear how we get there. Chemostats are good to a point, to about 0.025, a growth rate of 0.025, but to go lower, it's a little bit tricky to have a nice controlled system. So we use a retenstat cultivation, which is very similar to a chemostat, where you have full cell retention. When you combine that with a possible glucose feed gradient, you can transition to a point where the substrate uptake rates basically are enough to satisfy maintenance, but not to grow. It's very different to starvation conditions. The cells remain metabolically active. And so I'm lucky enough to work with Karina, who performed the retenstat cultivations, which I could use my data for. And at the start of this year, we managed to bring together 2 separate stories where Karina performed retenstat cultivations of a secreting strain, specifically this delta flow 8 VHH producing strain, VHH expressed under a glucose limit inducible promoter. And she was able to demonstrate that at very slow growth rates, and we're talking doubling times of 50 to 60 days, there is still active VHH secretion and production. As part of the study, she also looked at biomass compositions, which I was able to use constraint-based methods to understand how metabolic fluxes change at these slow growth rates. And you can find both of those studies here. Also, as part of that study, RNA was extracted, and that basically allowed me to start with identifying long non-coding RNAs. So there's 4 different sampling points during the red tent stack cultivation, as well as a sampling point from a chemostat at a growth rate of 0.1. So this is approximately a 250-fold range in growth rates, and a good starting point. I implemented a long non-coding RNA prediction pipeline, which is basically built up of taking the reads, mapping to the genome, comparing this assembled transcriptome to the reference genome, and at which point we can identify that there's 141 potential long non-coding RNAs. Implement a couple of different filtering steps, which look at those transcripts and see whether they have been reported in different RNA databases previously, whether the translated versions of those transcripts contain any potential protein domains, removing those transcripts that are not reasonably expressed in all samples, as well as using a couple of different coding potential tools. We're then left with 106 different unique long non-coding RNAs that pass these filters. As a final stringent, very conservative filtering step, they're classified with respect to their protein coding genes, and I removed any long non-coding RNAs which were not multi-exonic, not more than 150 base pairs from their neighbor, and not antisense. So I'm actually left with only 52 long non-coding RNAs, 8 of which are multi-exonic. It's quite strict, it's quite conservative. But at this point, there's long non-coding RNAs. Do we see any difference in their expression levels according to growth rate? So perform a gene level based count of our whole transcriptome, and what we'd hope to see in this experiment is that there was a separation of the sample points according to the growth rate. And when you perform a principal component analysis, which basically looks for the largest source of variation in your data, what I'm happy to see is that along the first component, the x-axis, we have a clear separation of colour, which is here. The sample points are proxy for the growth rate. And that is the same when we look at all transcripts, and that's also the same if you look at just long non-coding RNAs, which suggests that growth rate is also a large source of variability here. When looking at between the fastest and the slowest growth rates, we see that there's 20 long non-coding RNAs that were upregulated, 2 of them are downregulated. So again, just confirming, we have long non-coding RNAs, there's growth rate dependent expression. How do I try and work out what their function is? So I decided that I can't look at interactions with proteins, I can't look at interactions with peptides, but maybe I can try and look at the relationship with other transcript levels from this data. So I looked at the correlation coefficient of the expression levels of the long non-coding RNAs with all other genes. And what we see is if we look at just all, which is here in red, we have a roughly bell-shaped distribution centered at 0. But if we look at long non-coding RNAs and genes that are nearby it, then we see that there's a shift towards a more positive distribution, which may suggest that long non-coding RNAs positively affect their neighbors, but it could also just be that there's an open chromatin conformation which affects their expression. One of the other methods is this in-trans interaction by forming triplexes. Used a couple of different tools to predict interacting partners between a long non-coding RNA and different protein coding genes. And when we look at the correlation coefficient here, we see that there's more of a bimodal distribution of both positive and negative correlation, which actually suggests that a long non-coding RNA might be expressed and interact with duplex DNA of a protein coding gene, which might boost or may up or down regulate this target gene. So There's evidence that there's both cis and trans functions of these long non-coding RNAs, but yes. Beyond that, we wanted to see whether of these long non-coding RNAs, we have some idea of function. We see that there's growth rate dependent expression, but I also wanted to verify them experimentally and see whether if we perturb their expression levels, whether we see any difference. So I decided to use a CRISPR activation strategy. I was targeting target sites very close to the target, to the transcript start site of these new long non-coding RNAs. And basically this CRISPR activation strategy was released from our institute. Uses a deactivated Cas9, a scaffold guide RNA, which interacts with an MS2 binding protein fused to an activation domain. When you target this to near the target transcript start site, hopefully you will increase expression of your transcript. I wanted to go for an in situ strategy because it might be localization dependent, where the long non-coding RNA is. I integrated this whole cassette into a wild-type strain as well as a protein-producing strain, specifically the one from the retenstats. I tested at least three guide RNAs for 9 different long non-coding RNAs and compared this to a non-targeting control, which contained everything, including a guide RNA, except this was predicted to not interact with anywhere in the genome. When we look at the maximum growth rates on glucose, we didn't see any significant differences, but then I also wanted to look at protein production. So I used a 24 deep well plate fed batch-like screening assay, using enzyme-controlled glucose release, and at the end of 72 hours, measured the wet cell weight and titer via capillary electrophoresis. Now, when we compare to the non-targeting control, over here on the left, we see that the relative wet cell weight was not affected. When we look at titer, we do see that some of these different strains, some of these different target sites, lead to a reduction in titer and subsequently the yield. And here you can just see along the x-axis are the different strains, the different guide RNAs, and the dotted vertical lines just separate the different long non-coding RNAs. Some of them have 3 sites, some of them have 5, etc. And yeah, so we can see that some guide RNAs, when expressed, lead to reduction in titer. I needed to verify whether this is because of the long non-coding RNA being overexpressed, so I will just dive into looking at 2 of those long non-coding RNAs here. On the top, you can see the screening results again. However, along both plots, the x-axis, you can just see where the relative position is of the target site for the CRISPR activation cassette relative to the start of the long non-coding RNA here. And just on the opposite strand, you can see the nearby coding gene. And on the top, you've just got the relative yield. On the bottom, I have the log 2-fold change of the long non-coding RNA transcript level. And you can see that here we do indeed have overexpression of this long non-coding RNA, which does seem to match up nicely with where we see a big reduction in yield. I sent these strains off for sequencing and compared these strains compared to the non-targeting control, these values here. And quite nicely, we can see that, yes, our long non-coding RNA is differentially expressed, as in the qPCR, and we also see that there are many other genes that are also affected. Some of these are predicted to interact with this long non-coding RNA via triplexes. When I look at the enrichment of the genes, I see that those that are downregulated are involved with transporters, transcription factors, but there was no specific enrichment in the upregulated ones. Looking at a different long non-coding RNA, we see a similar story. Interestingly here, we do see upregulation of the long non-coding RNA transcript, but no real difference in the yield. Here, it's not quite as high, but we do see a big reduction in the VHH yield here, suggesting that overexpression of this long non-coding RNA is one of the explanatory factors. Sent this off for sequencing too, and I was really very happy to see this result, that the most differentially expressed transcript was our long non-coding RNA, which is right up here, as well as a couple of different interacting partners. The same was true for target site 2. And then for target site 4, we still had significant overexpression, but it wasn't quite as high as the other ones, the same as in our qPCR results. And also those predicted interacting partners were also differentially expressed. In this case, we just saw that some genes related to peroxisome, microbody, and the ribosome were regulated, and some transporters were down-regulated. But these are all extremely broad terms. So even though we've managed to demonstrate that we can annotate long non-coding RNAs, there is growth rate-dependent expression of them, and there are maybe some ideas of what their function are. Even if you do perturb their expression levels and you do see differences on a physiological or transcriptional level, it's still really quite hard to understand why. And yeah, basically with that, I'm at the end of my talk. And I need to say thank you firstly to Victoria Trevarts, who is a technician in the CD laboratory, who's been really important for me during my PhD, but specifically in this project. She supported me as well as Karina Rebnega in the original Retenstat cultivations. Of course, to Brigitte Gasser, my supervisor, and also to Krishna and Batset Said, who've been working with me at points during this long non-coding RNA project. Also to the funding partners, to the Christian Doppler Society and Lonza, and of course you for your attention.
Thank you for a very, very, very interesting presentation. So I was wondering whether you can comment a little bit more on where the location is of these long non-coding RNAs in the genome. Are they really within, like in between of the genes? Are they in, so maybe if you can give a bit more.
There's a real distribution. If I highlighted that there was that stringent filtering step, that removed a lot of long non-coding RNAs which are close to protein coding genes, maybe about 30 base pairs away from one. If I looked specifically at that level, so the 106, I think I said, if I look at that, then I have a real distribution across the whole chromosome. I see that there are some localized in repetitive regions, so there are some that are very close to flocculin-related genes, but in general, we identified long non-coding RNAs representing all classes. We identified some that are fully antisense and contained within a gene, some that are antisense and overlapping, those that are convergent, so you have two on the same strand coming towards each other. We managed to find some of all of them. The most common ones are intergenic, which would be the easiest ones to target. But actually those long non-coding RNAs that I targeted are a real mixture of some that are antisense.
Hey Ben, nice talk. Question regarding your growth rates, right? So 60 days, doubling time. Do you think there was some other interference with, such a low growth rate and, the maintenance demand of the cells under those conditions?
There's massive effects. It affects everything. But specifically to the long non-coding RNAs?
Yeah.
Okay. Do I think that there are confounding factors that would affect my...
Correct.
Probably. But I'm in the lucky position that I don't know what they are.
Okay. Well,
but there are definitely some. However, the final 3 sampling points from the Retenstat, their growth rates are more close to each other. They're more similar to each other. The other 2 growth, so the 0.1 chemostat and the 0.025, already there's a 4-fold difference between them, but then there's a big jump before the next lot of samples. So 2 samples represent a growth rate above 0.025, and the other ones are all below 0.02. Yes. But in terms of affecting the transcript level, I can't exactly say that the only source of variability we see is growth rates, because that's a very broad term to try and isolate as growth rate specific. We're also producing a recombinant protein with this strain, and the effect of that combined with the growth rates also affects stuff. Yeah, it's really hard to say.
Okay, thank you.